Hello, I'm Rick Roush. I'm the Dean of the College of Agricultural Sciences. We're excited to have you all here with us today for our College Connections webinar. These webinars are designed to give you a unique inside perspective of the diverse programs, people, priorities, and partnerships within the Penn State College of Agricultural Sciences. We are recording this session. You can find the links to this and all past College Connection webinars, as well as registration information for future events, on our college website by searching College Connections. Please note, it does take a few days to edit and post the recordings. Today's session is Discover the Microbes Within, and I'm joined today by Dr. Seth Bordenstein, who's the director of the Microbiome Center at Penn State, and Dr. Sarah Bordenstein, who's the associate research professor and director of Discover the Microbes Within, a program through the Microbiome Center. After the presentation, we will have a Q&A session, first addressing those questions that were submitted during registration. If you have questions during the presentations, please enter them in the Q&A link, not the chat, because it's easier for us to track them there. Before we dive in, I'd like to take a quick minute to introduce our presenters. Dr. Seth Bordenstein is a recognized thought leader and scientist who studies the centrality of microorganisms to the biosphere and human health. He has peered into the world of microorganisms that dwell inside plants and, that dwell inside animals for the past 25 years. His research specialties span the extraordinary utility of microbes to control mosquito-borne diseases, the secrets of microbiome diversity in the diversity of humans, and the major trends of host-associated microbiomes across the animal kingdom. He is a director of the world's first One Health Microbiome Center, the Dorothy Fear Scott and J. Lloyd Huck Endowed Chair in Microbiome Sciences and Professor of Biology and Entomology. Dr. Sarah Bordenstein is the director of the Discover the Microbes Within, in which she develops and disseminates educational resources related to microbial symbiosis, biodiversity, informatics, and biotechnology. Featuring research topics and cutting-edge technologies from the One Health Microbiome Center, the program translates complex laboratory protocols into approachable, classroom-ready curricula that are being used to train the next generation of the STEM workforce, that is, science, technology, engineering, and math. The project spans more than 20 countries and 45 states, engaging thousands of high school and college students each year. Sarah is the recipient of numerous education awards, including the 2023 T.H. Huxley Award from the Society for the Study of Evolution. All right, with that, take it away, Seth and Sarah.
All right, thank you very much, Rick, for that introduction. I am so glad you are all joining us today and spending a little time with us. There is a good amount of brain candy that I hope you'll enjoy as you walk through two presentations today from Sarah and I. I'll cover the first 15 minutes and then she'll cover the latter 15 minutes. So I'm pleased to represent the One Health Microbiome Center here at Penn State. We are the first One Health Microbiome Center in the world. I want to clarify to you why I think we've got something special, why I think our university is well served by this center, and what kind of impacts the science of the microbiome and our faculty and trainees are making and will make in this relatively challenging 21st century that will illuminate a little bit more. So join me on this journey as we go through, and then we'll hand it over to Sarah for how we're reaching out through education and outreach to educate the world, really. So the One Health term is a relatively rising term in the life sciences, planetary sciences as well. In our vision of this term, One Health really means the interconnectedness of these three domains of health: human health, agricultural health, and environmental health. And each one of these essentially domains or ecosystems, the agricultural ecosystem of crops and livestock, or the environmental ecosystem of our world's oceans or terrestrial environments, or our own health ecology and human health, essentially are all dependent and reliant on microbiomes. That is, the community of little tiny life forms, microbes, viruses, bacteria, that can flow through these environments and interconnect them in rather fundamental and important ways. So we live on a microbial planet. Everything is colonized by microbes, and these three domains of life have their own microbiomes as well that may be shared to determine the risk of health and disease in each one of those particular pillars. Now, the One Health Microbiome Center is uniquely positioned to tackle this kind of grandiose vision that we live in a microbial world and the health of these domains will be influenced by microbes. Because we have 115 faculty scattered throughout the university, 42 departments are represented by those 115 faculty in the center. And there are 500 total plus members, which goes from the faculty all the way down to the trainees, including undergraduates, that participate as active members in our microbiome center. We have a weekly email newsletter. So for those of you affiliated with Penn State University and would like to hear more and get interested from what you hear today, please do reach out to us and we can get you on the newsletter. Ultimately, our goal is to really define and be one of the great microbiome centers of the 21st century. And we really do mean that because of our wide-scale depths of expertise and our influences in not just one of these domains of humans, ag, or environment, but in fact, all of them together because we have such a great depth and team here. So let's start with us, the human microbiome. When you look at your own body, you need to think of yourself not just as human, but really half human and half microbial. And that's because there's a whole consortia of microbes that live in and on your body. And in fact, each body site has their own unique set of microorganisms that live there. Your skin microbes are different from your oral microbes, which are different from your gut microbes. In terms of numbers, the vastness is hard to understand because there are more microbes in your mouth alone, your single mouth, than there are people on the planet. That's an extraordinary number to think about because your whole body is covered in microbes. There are estimated 40 trillion microbes, bacteria, and other tiny life forms that live in and on you. And from those 40 trillion microbial cells, we have about a 500 to 1,000 bacterial species living in these various body sites. Now, the microbiome is rich with genetic potential. There's, in fact, if you take all the microbes and all their genes and ask that to compare that to the types and number of genes we have in our own human genome, there's 100 times more genes in the human microbiome than there is in the human genome. And these genes can play extraordinary roles throughout our lifetime, whether it's immune system maintenance, digestion, nutrient acquisition, metabolism, and then pathogenesis, of course. Microbes are most often helpful or harmless, but we often hear about the pathogens, the few microbes that actually make us sick. Now, another perspective is our DNA from a human species perspective is extraordinarily similar between individuals. 99.9% human genome similarity between any two individuals on average. If you look at the microbiome, though, there's up to 80% variation between people in the types of microbes that they have. That is, consider the types of bacterial species that they may have in their gut. Your microbes are more different to the person you might be sitting next to or having lunch with than your genomes are by an amazing capacity. And that means what makes us potentially physiologically unique and physiologically different, whether we're susceptible to cold viruses, whether we digest milk or not easily, is potentially related to which microbes we have and what makes us different. And of course, I think a lot of us now know that many diseases have a correlation with microbiome changes in our body. That doesn't mean they're causal, but it does mean that there's a large set of associations that science is working on right now to see if there's any causal relationships and how we can dial those relationships in favor of having positive influences on our own health. So we live in a microbial world, and we then have our own microbial lives. This is a rule of the biosphere. For all life forms, we are living on a planet dominated by microbes. So there's no sterile environment wherever we look on the planet. In our own human bodies, as we westernize into cultures that, let's say, are more urbanized, more commercial, less organic, more medication, we're also seeing in the human species there's a massive decline in microbiome diversity as we westernize our culture. So we're losing essentially those microbes that we formerly had where we weren't living in such an industrious time. And interestingly enough, as our microbiomes take changes and decline in diversity, that is, we have fewer species of microbes than we used to have, it's interesting that there's a rise of chronic diseases also in westernized countries, metabolic syndrome, cardiovascular disease, and there's also a rise of antibiotic resistance. And these two things may in fact be correlated from a human health perspective. This is some beautiful patterning done from a 2002 study in the New England Journal of Medicine that showed that humans have been very successful in specifically targeting infectious diseases because we have developed antibiotics to many of these, or vaccines. These diseases, single microbe diseases, have declined over time. But right at the same time span, there's been a consistent increase in chronic diseases that are much more complex to us right now, much more complex to treat as well, and have a microbiome association. And what I want to illuminate to you is that Penn State's University human health microbiome folks are all over this particular topic right now. So from Hershey's campus to the University Park campus here, we have scientists deeply embedded in many of these important topics that affect human health in westernized countries today. There's also an aspect of not just thinking about adulthood, but also thinking about childhood and how our early life experiences shape the microbiomes and our risk of disease as we develop into adults. There's this hypothesis, and essentially what it means is that there's an early critical window when we acquire our microbiomes from our family members, from the way we were born, and from our environments. And that early colonization could potentially have an outsized effect on how our long-term health and short-term health is determined. And that's because you're born sterile, but microbes colonize you and then train your immune system and train your physiological functions to do all the things that allow us to operate, because we couldn't exist without microbes. And you can imagine that the types of microbes you acquire early in life will influence the training of your immune system and physiology later in life. And so this offers an opportunity for intensive study as well. We have investigators who are looking at adult diseases as well as childhood diseases, such as the development of allergies and asthma. Now, from a more environmental and agricultural perspective, but also related to human health, the deadliest animal in the world is this particular mosquito, the mosquito species. And the reason is mosquitoes can kill up to 1 million people a year by the diseases they transmit when they bite us. And they can cause about 400 million cases of infectious diseases, many of which are viral diseases such as Zika and dengue virus. Now, as the climate warms, we have a risk of increasing environmental ranges for these mosquitoes. And as humanity tends to go on an increase in urbanization, we also create more breeding grounds, wet breeding grounds around people that could allow for more mosquito problems. So this is not something that's getting better. This is something that's getting worse. And so the mosquitoes will remain the deadliest animal for a while. Now, our lab and those at Penn State in the Microbiome Center also study a bacteria that essentially turns this deadly animal into a miracle mosquito, because these mosquitoes with the bacteria known as Wolbachia no longer will transmit Zika and dengue virus transmission. So the kind of work we like to think about is engineering the world's life in order to enhance our own positive outcomes. And in this case, we can change mosquitoes to become helpful mosquitoes by adding in this particular bacteria that stops Zika and dengue virus transmission. There's also a massive problem in agriculture that is going to relate to the microbiome, which is population increase is estimated to be about 9.7 billion by the year 2050. In order for us to accommodate that population increase, currently we're at about 8 billion people in the world, there has to be a massive increase in food production. It's estimated that there needs to be a 60% increase in food production in order to handle this. So that means that we're going to turn to our agricultural health and microbiome scientists to think about developing better ways to sustain good microbes and good output of these animals and plants, whether it's the types of feeds we offer to these cows in order for them to enhance their output and their physiology, or it's the types of microbes we put in the soil in order to grow better crops such as corn or tomatoes. How we cultivate the microbiomes of these larger visible life agricultural keystones will be central to essentially making better food production in an optimized way especially in a world that's warming and undergoing various types of climate stress. Related to climate stress, the oceans are warming, and we have experts in the One Health microbiome area who are studying exquisite animal microbe relationships in the sea. In fact, this is a squid that's fluorescent. You can see that glow, and it's fluorescent because it has a microbe inside of its organs that allows it to produce this glow. And it has served as a model for understanding animal microbe relationships in the sea in an exquisite way. Coral really take the prize. I'm sure many of you are aware of coral bleaching, but coral bleaching results because they are losing their microbes that allow the coral to do photosynthesis. And so these do photosynthesis, and these coral bleached ones do not. And that's because they lost an essential microbe. Microbes will be solutions for us in the 21st century, as you're hearing a little bit more about today. So at the One Health Microbiome Center, we really think of life as foundations is built in the microbiome, that our biosphere is completely dependent upon it, and that the microbes from the soil to our livestock and crops to our own bodies are all going to be relevant to critical solutions in the 21st century. Moreover, there may be flow of these microbes between the environments, from soil to crops, from crops to agriculture, from agricultural to insects, and then to humans. You can imagine how these particular systems are connected by the way microbes flow through them. Now, we're not just the only ones thinking big picture about One Health microbiome sciences. The National Academy of Sciences has launched what they've called a One Health Action Collaborative, where they've organized many governmental and industrial groups, CDC, the WHO, the USDA, the Department of Agriculture, et cetera, in order to prioritize thinking about microbial flow from one environment to another and how that poses threats to health of not only our bodies, but agriculture and environment. So to wrap up a little bit, I want to highlight what our strengths are, if they haven't sort of been illuminated already. At the One Health Microbiome Center, we offer the first and only microbiome sciences PhD degree in the world. We believe that this is a field that will transform the future. We're seeing that already from our science, and we got to accredit it and affirm it with the degrees we offer from Penn State. So we are doing that right now. We are also one of the largest and most active microbiome centers in the nation, if not the world. As we've highlighted, we have excellence in these 3 domains. And we also offer a top high school science education lab series that Sarah will talk about to you in just a second. And so with that, I think I'll wrap up and leave this directly over to Sarah, as she'll pick up on this particular outreach and science education she does. If you have any questions, put them in the chat box, or we'll take them at the end when Sarah's done with her particular section. Thank you, everybody.
Okay, so thanks, Seth. So following Seth's grand overall vision of the One Health Microbiome Center, we wanted to briefly introduce you to our flagship education program, Discover the Microbes Within. So through the program, we aim to transform STEM education so that it better represents real-world science, which in turn is going to prepare our next generation STEM workforce. And again, by STEM, we're referring to science, technology, engineering, and math. So Discover the Microbes Within has a fairly comprehensive representation throughout the US, including about 45 states and the Washington, D.C. area. And here you can see that our partners are pretty evenly distributed between high school and post-secondary education. And at the post-secondary level, we have participants all the way from 2-year biotech and community colleges, all the way up to liberal arts colleges and larger research universities. The Discover the Microbes Within platform was really developed to provide students with an experience to become scientists by encouraging discovery, inquiry, critical thinking, and curiosity with hands-on problem-based learning. And hold on just a second. Oh, there we go. And so this is a deviation from the old-fashioned way that science classrooms used to be taught, where it was all based on cookie cutter labs and just basic memorization of facts. Instead, we engage students with nature, and we provide them with cutting-edge technologies so that they can really begin to address real-world questions. And ideally, at the end, we can facilitate student contribution of new scientific data on biodiversity and symbiosis back to the scientific research community, as well as local community stakeholders. This is particularly relevant because STEM careers represent about 23% of the US workforce, and notably about half of those don't have a bachelor's degree. In addition, the US Department of Commerce four-year strategic plan prioritizes research and advances in biotechnology. So not only does this suggest that we must train and empower our K-12 educators, but we also have to encourage the development of biotechnology programs in our local school districts and communities. And as Seth kind of alluded to, we would strongly agree that biotechnology programs must include the microbiome sciences. And so in many cases, as these biotechnology programs are getting up and going in schools, Discover the Microbes Within is the introductory unit in these programs. So due to its success, we are starting to see through word of mouth the wide-scale global distribution of Discover the Microbes Within. We currently have about 20 partners across the globe that have already implemented the program or are interested in implementing the program. And as Seth mentioned, one of the core focal areas of our research lab is Wolbachia. And so we started this program about 17 years ago with a focus on one microbe, and that being Wolbachia. As you can see here in the colorized image, the yellow cells are Wolbachia. Wolbachia is an obligate intracellular endosymbiont that lives within the cells of insects. And most importantly, it's a model system for exploring symbiosis and host microbe interactions. So because Wolbachia lives within the cells of insects, and there's over 1 million named insects on the planet, with many more left to be discovered, Wolbachia is in about 40 to 60% of all of these insects. So that really presents just an astounding, overwhelming opportunity for students to contribute real data and ask really meaningful questions about their communities. Students can explore invasive species, household pests like the bedbug, agricultural pests, pollinators, vectors, and more, all within their classroom. And it's important to note that Wolbachia, while it does live within the cells of insects, it does not infect humans. So that makes this one particular microbe really safe to study in the K-12 classroom. Okay, so why would we want to focus on microbiome? Why would we want to start with a microbiome project on Wolbachia? That's because it has a really fascinating biology. So Seth already alluded to the fact that it can inhibit the transmission of diseases, but Wolbachia also acts as a reproductive parasite. So Wolbachia is maternally transmitted. That means that this bacteria, this microbe, passes from mom to offspring. So every insect gets its Wolbachia infection from its mom, not its dad. Therefore, Wolbachia has had to evolve these really cunning strategies to enhance for the numbers of females in a population. So in this example, let's look at the Aedes aegypti mosquito, for example. In the wild, it doesn't naturally have Wolbachia, and so we would expect to see both male and female offspring. However, in the roly-poly, which does often have Wolbachia, Wolbachia induces what's known as feminization, where it turns genetic males into fully functional phenotypic females. In ladybird beetles, for example, Wolbachia can induce male killing, where it selectively kills the males, allowing the female offspring to survive. And in spider mites, Wolbachia has completely eliminated the need for males. And now these spider mites can reproduce asexually via parthenogenesis, whereby the females are essentially creating daughter clones of themselves. So as you can see, in each of these, Wolbachia has really cunningly allowed for the success of females. That way it can be passed on to the next generation. The primary focus of our lab is a fourth phenotype called cytoplasmic incompatibility. Now this, while it may not associate with sex ratio as much, it actually allows Wolbachia to rapidly spread throughout a population. And this is because Wolbachia can modify the sperm of males such that that male can only successfully reproduce with a similarly infected female. So in this cross, this is a standard genetic cross, you can see that the open circle represents an uninfected individual, an uninfected male. If he mates with an uninfected female, so they don't have Wolbachia, their offspring are also not going to have Wolbachia. However, if he mates with an infected female, a female that does have Wolbachia, because she is transmitting that Wolbachia, all of her offspring are going to have Wolbachia. Likewise, if she mates with an infected male, all of their offspring are going to have her Wolbachia. However, if this infected male mates with a female, she doesn't have that Wolbachia that can rescue the sperm modification, so their offspring die. However, if the same male were to mate with a female that has his same Wolbachia, his offspring will survive. So as you can see, over time, we have an enrichment for Wolbachia infected individuals in a population, and it can rapidly spread. So going back to what Seth was talking about Wolbachia being able to inhibit the spread of mosquito-borne viruses. So if we have Wolbachia in a mosquito, as he said, it can inhibit the transmission of the virus. And one way to look at this is, for example, when we think about dengue or chikungunya or Zika, if a mosquito doesn't have Wolbachia, it bites a person with the virus replicates within that mosquito, goes on to bite the next person, and will transmit the virus to that person. However, if that mosquito doesn't have Wolbachia, unfortunately, it still bites the first person, but it inhibits the transmission of that disease to the second person, so that person does not become infected with that virus. So this is really amazing. However, it's really not feasible for us to go out and singly inject every single mosquito out in the wild with Wolbachia. But if we take what we know about cytoplasmic incompatibility, we can raise mosquitoes in the lab that have Wolbachia. We can go out to communities where Wolbachia doesn't naturally exist. Releasing these infected mosquitoes, they essentially will replace that population with mosquitoes that have Wolbachia but can't transmit disease. Alternatively, if the mosquito is invasive, or maybe if it's causing some type of environmental or ecological issues, we can instead only release those males. Those males will mate with the uninfected females, and it'll crash the population because they won't be able to successfully reproduce. So depending on your needs, we can utilize cytoplasmic incompatibility to either replace or suppress an insect population. So obviously, taking it back to the students and back to education, when they learn about this biology, they are really excited about Wolbachia and how we might be able to use it to handle problems that are in their communities. One thing that I often get asked by students is, well, does Wolbachia infect the spotted lanternfly? Could we maybe use that to control the spotted lanternfly? And when I Google Lycorma delicatula, which is the scientific name, and Wolbachia, you'll see that some of the top hits are actually from K through 12, from high school students. And these high school students have participated in the Wolbachia project. They've gone out and collected a spotted lanternfly, given us a little bit of information about its behavior, its taxonomic ID. And then they also upload their experimental methods and their results. So they're going to upload images of their gels and all their raw data so that me as a scientist can go in here and quality check their data and see if I agree with their results. They also sequence the DNA from both the Wolbachia and the arthropod, upload this. So again, community stakeholders can reference these DNA sequences as we begin to study the spread of the spotted lanternfly and maybe learn a little bit more about the Wolbachia association with the lanternfly. Importantly, each one of these entries has a unique URL. So these students, whether they be high school or college, can use it on their college applications or job resumes as evidence that they have used and know how to perform these skills. Alternatively, I've had students look at Brood-X cicadas. And again, if you Google the cicada with Wolbachia, you'll see that some of the top results are from high school students. Not only did these students enter it into the Wolbachia project database, but they also went on to publish their results in a journal for young scientists. Okay, so what is the Wolbachia project? So the Wolbachia project is essentially a 5-part lab series that covers biodiversity, biotechnology, and bioinformatics. Students will come up, they will ask, it's really unimaginable some of the questions that they can ask about their environment, where they go out and collect insects. They bring the insects back to the lab, extract DNA. They amplify the DNA using PCR. So this is similar to like the PCR test that we might do for COVID. So it's really applicable to the healthcare sciences. And they can use gel electrophoresis to visualize the DNA. Finally, they can sequence the DNA and apply bioinformatics to identify their organisms and also look at evolutionary relationships of these organisms with closely related species. And I really want to highlight that we are dedicated to making this free and accessible to all students, regardless of your geographical location or socioeconomic status. We provide the free reagents. All the curriculum is available under a Creative Commons license. So that way teachers can take it and modify it to best fit their classroom needs. And we really have training videos, and we want to help teachers and citizen scientists as they explore their local communities. And as I mentioned, we have a Wolbachia project database that features both positive and negative data. Both are equally valid. When we're looking at host microbe interactions, we really want to know, is the microbe there or isn't it? Students can upload this to our database. In the last 2.5 years, we've had almost 1,500 posts from nearly 1,000 students. And so moving forward, thinking about the whole microbiome in general, we've been pretty successful with the Wolbachia project. I mean, it's a fascinating biology. But while Seth and I could study this for the rest of our careers, we realized that a lot of people are interested in the complete microbiome. Teachers really want to know about the environments in which they live, and they want to be able to go out and ask their own questions about local microbiomes. So we are currently taking our curriculum and we want to modify it so that we can enable teachers, students, citizen scientists to be able to look at environmental and food microbiomes by doing many of the same techniques. And then at the end, instead of just sequencing a single gene, they're actually going to use next generation sequencing to look at the entire microbiome. And we will teach you how to do microbiome data analysis. So if you're interested in learning more about this and kind of keeping up to date with how we progress, we are working with collaborators throughout the One Health Microbiome Center to develop this curriculum. And we just want to say that, I mean, we are living in a microbial world, and we thank you for being here today. We thank all of our collaborators in the Microbiome Center and the thousands upon thousands of teachers and students that every day are asking such amazing questions and making new discoveries. We can't wait to see what's ahead in the world of the microbiome sciences. And we encourage you to reach out to us. And if we can, drop in the chat box all of our social media and website details so that you can further follow us and keep up to date. Thank you.
That's fantastic. Oops, there we go. That's fantastic, Seth and Sarah. Sarah, one question that came to mind for me right off the bat was, when you say apply bioinformatics, what does that involve?
Yeah, so we currently are looking at Wolbachia and insect barcoding. So we employ a lot of insect barcoding in our project. Bioinformatics currently is where they will sequence the DNA, where they will target either a barcoding gene, the cytochrome oxidase from an insect, or a barcoding gene from Wolbachia. They can then take this DNA and we will help you to sequence that DNA, and we'll provide you with the sequence. You can then use that sequence and compare it against the National Sequence Archive, which is NCBI. It's a freely available tool that has all the DNA sequences from everything that's ever been sequenced available. When you reference your sequence against that, you can identify the identity of your organisms, and you can also begin to build phylogenetic trees. And we rely upon free websites and free tools to do all of this, and we have instructions on how to do that online.
Fantastic. Okay, so we have a number of questions that were submitted during the registration process, and a few in Q&A already. So, so first off, for Sarah, the question was, how can I introduce this information to my students? What are the strategies, projects, et cetera, that we can use?
That's a great question. So I would highly encourage you to reach out to me directly by email, or you can visit wolbachiaproject.org. There you can download all of the lab activities, you can watch the video tutorials, and you can also request the free molecular reagents for us. There's also tools on how to get started on the website. I would encourage you, if you're really new to this and you're just getting started, start with something easy, like looking at the biodiversity of your backyard. Just go out and have fun and collect. And then once you become really familiar with the skills and the techniques, then you can begin to ask some really meaningful questions, like looking at the invasive species, and start to accumulate that data. We have some teachers that work with local mosquito commissions, for example. They get funding from the mosquito commission to go out, barcode the mosquitoes, tell whether or not they have Wolbachia, and report that back to the commission. If you think about it, as scientists, we can't do this all alone. We need help. And so by training an army of students, we have thousands upon thousands of people in your community that you can call upon to get this information for you.
That's fantastic. So how can you beat that as an offer?
Yeah.
You want to get involved, right, Sarah? You can see she's pretty excited about this. I just want to mention that contact details for the Bordenstein lab and so forth are in the chat. So if you want to, you can flick there and you can find some of the contact details. So Seth, the next question suggested was, can you suggest a book for further reading on microbiomes?
Yes. So one of my favorite pastimes as a kid was watching nature shows on the weekend and turning on PBS and maybe dialing to David Attenborough and telling us a great story about the visible world. There's a wonderful book that does the same thing, but for the microbial world, called I Contain Multitudes. I Contain Multitudes, by Ed Yong. And he has written for the New York Times. He's a current writer for The Atlantic, and basically gives us the PBS David Attenborough show in text for microbiomes. You might even find a few familiar names in there that you've heard about today.
That's great. Okay, so next question was, and again, maybe for Seth, how did the first microbes arise on Earth?
Okay. All right. This is a very big question that is difficult to know for sure, but scientists are looking at essentially two hypotheses for how microbes first colonized and got here. The first one is sort of that prebiotic soup where there's a mix of ingredients on the planet that could eventually come together and form a cell, what we actually call a protocell in science, which is getting a cell membrane and getting some genome inside of that. And without going into a lot of the details, scientists have actually been very effective at recapitulating the conditions where small chemical molecules can group together to form what is a protocell, or this near cell life form. The other hypothesis for how microbes got started here was they were transferred on, let's say, meteorites from one planet to another, eventually hitting Earth, and then that brings in water and microbes with it. One thing's for certain, if we're going to find life anywhere else in the universe, it's going to be microbial first, because that's the story of Earth. Microbes colonized Earth 3.6 billion years ago and basically have been here dominating since. And all complexity of life has come from microbes. So if we're going to see life elsewhere, it's from microbes. And if they're traveling on meteorites, which is shown to be possible by experimental tests, by embedding microbes under rocks that are vectored at high speed, they can survive that. It's possible that they're moving between planets. So those are the two current hypotheses and a bit of evidence for both of them.
Which still begs the question of how to get started on the other planet too. So
yeah, it might be a similar story about bringing all these ingredients together that scientists have been really good at. Yeah,
that's great. So the term biofilm has come up in some papers about water quality and waterborne pathogens that I've read lately. If I understand the term correctly, they can act as a sort of aggregators and protectors of bacteria and other tiny organisms. Are there any connections between biofilms and microbiomes? Could a biofilm become considered a microbiome? Thanks for any clarification or edification. Sarah, you want to take a crack at that one?
Sure, yeah. So yes, definitely a biofilm is one type of microbiome. So a microbiome is a community of microbes that live together. And the biofilm means that the community of microbes has attached to a surface, it's expanded and grows, sometimes in an uneven manner, that allows channels of water and nutrients to flow through. And often you'll see biofilms being layers, like mats, different layers of mats of microbes that grow one on top of another. Unfortunately, some of these biofilms are associated with human infectious diseases. And so if a pathogen is part of a biofilm, it can be sometimes hard to treat that particular disease because antibiotics and medications can't penetrate the biofilm. So we can see this, for example, in tonsillitis. A more severe problem might be the Pseudomonas biofilm that's associated with the lungs of cystic fibrosis patients. So I would say that, in these cases, we really need to understand more about these specific biofilms and the interactions between the microbes, because it does have consequences for human health and how we treat disease.
That's great. So next question was, how do we test a microbiome? Who determines what a healthy one is?
Yes. So the testing industry is, I think, still in development, but there's two projects that I can recommend for self-sampling. One is the American Gut Project, where they'll send you a kit for about $99 that you would then swab, let's say, a fecal sample or whatever it is that you want to sample, put it back in a tube with a storage buffer, and then send it back to American Gut, and they'll sequence, profile it, and give you a report back. There's another company, commercial company called Viome that could do something similar. So American Gut or Viome. None of these are at a stage yet, and the science really isn't at a stage yet that'll give you medical advice. In fact, the government doesn't authorize these companies to offer you medical advice on whether you have a healthy microbiome or not. So it's more of a profile and snapshot in time, just to learn a little bit more. You can also sample your pets. There's a company called Animal Biome. If you want to do your kitty or your Snoopy and sample what's going on with them, maybe if they're having bad issues too, maybe you would potentially learn something by getting data and doing research on your own. Healthy is a personalized outcome for your pet and for each one of us. And healthy microbiomes kind of is put in that context, that it's difficult to say that everybody should have this same healthy microbiome, because that's clearly not the case. Why? Your genetics are different. You grew up in a different environment. Your immune system was trained in a different way. So healthy really becomes personalized from a microbiome perspective to each one of us. And there won't be a silver bullet sort of reasoning. Now, there might be likelihoods based on population averages that if you have this bacteria, you might be predisposed to getting an infectious illness if it breaks out. But on average, consider your healthy microbiome one that's specific to you.
Right. So, Sarah, another one, can microbes help restore a soil that's been abused?
Great. Thanks for that question. Yeah, definitely microbes can help. I think what Seth was saying about a healthy microbiome for a person, the same can be said for different environments. So different types of soil might have a different type of microbiome that they're associated with. We typically use the term degraded rather than abused, because we see a decline in the physical and the chemical and the biological properties of the soil. So there's also a reduction in microbial diversity, where you'll only find in these degraded soils a few species of bacteria and fungi, for example, rather than really complex, healthy soils that include bacteria, fungi, archaea, protists, lots of different microbes. The soils can become degraded by contamination or the buildup of excess salt or salinization from agricultural processes, mining, and we can combat all of this by microbes. But in addition to the microbes have to have substrates in which they can grow and eat. So in addition to adding the microbes, we have to add organic matter to these soils. You'll often see farmers implement this into their farming practices by doing crop rotations. Maybe you'll notice the farm down the street, it might go from corn to soy to cover crop. This can influence the organic matter. They can also add compost and manure. They can do, reducing tillage, and all of that will allow the microbial or the microbiome to flourish and really enrich in diversity, because it has this organic matter to feed upon. It can recycle the nutrients and really stabilize the soil. Also, when soil is degraded, so maybe you're thinking of mining, or oil spills, where we have chemicals in the soil that are prohibitive to plant life, we can bring in the microbes that are able to degrade these certain chemicals. Again, we also have to have the organic matter for them to feed upon. But once the microbes begin to degrade the chemicals, they start to stabilize the pH. And then this begins to enhance plant growth by seeing more nitrogen fixers in the environment, for example. So yes, microbes are key, but it's really, when we think of the microbiome in general, it's really this association of everything that's involved.
Thanks, Sarah. So the next question was, what differences exist between how your gut microbiome reacts to non-GMO and GMO corn? Well, it happens, this is something I've worked a lot on, so I offer to take a first cut at it. So just to put everybody in the frame of the history of this. So genetically modified corn was introduced in 1998 with one gene from the bacterium Bacillus thuringiensis. So this is or BT. So this is a bacterium that was discovered in the 1930s to have insecticidal properties. And it turns out they make toxins that are very specific to insects that work in insects because they interact with the very basic gut of insects and are not effective against mammals, for example, because we've got a more acidic gut. So it's very specific. In fact, some of these BT toxins will only affect particular individuals in an individual family of insects. So they can be quite specific. So over time, after the first corn crops, other BT genes have been introduced to control caterpillars and also corn rootworms, which are beetle larvae. Also a gene that produces a plant model enzyme that is relatively insensitive to the herbicide Roundup was introduced. And it also produces another protein. So all these things are producing the active material from the GMO is a protein of one kind or another. And they basically get broken down in our guts. So first off, a lot of whatever we'd be exposed to in corn gets processed out anyhow. So in order for you to even get one of these proteins that's relatively intact, you'd have to eat something that wasn't processed very much. About 90% of the corn in the United States is GMO in one way or another, but a lot of sweet corn varieties and so forth you might eat aren't. So your chance of getting exposed to the active protein is pretty small, but even if you do, there was abundant evidence when these crops first went through the registration process in animal feeding studies to show that they break these three different kinds of proteins are broken down just the way any other proteins are, they're just chewed up. So the likelihood of having any impact on your microbiome is pretty small. And so that anyhow, it's probably unlikely that's one of the biggest problems we have to worry about. What do you think, Seth, about covered
That
all?
says it all. That's perfect. They just kind of pass right through. It's good to study it, and we actively keep monitoring it, but there's no evidence that GMO corn affects our gut microbiome.
Yeah. So the next question was, please comment on the use of pesticides versus industrial agriculture on the soil microbiome in human gut. So we talked a little bit about human gut already, but what about everything that's happening out in the soil?
Yeah. So, when I hear questions like this, I envision, can you imagine like a whole group of 4-H students going out and studying this? I mean, being part of the 4-H curriculum. I really think we don't know enough about pesticides in the soil microbiome and human gut. From what we do know, we have to remember that there are many different types of pesticides. So there's pesticides that will target weeds, the herbicides. There's the ones that control fungi. There's the ones that protect against insects, like the insecticides. And each one of these has a broad class of different components. In the US alone, there's about 800 different pesticides that are registered for use. So, when you read the news headlines and you see pesticides do this, I wouldn't jump to conclusions that all pesticides do that, because we have about 800. And each one of these can have different impacts on the soil microbiome. Some of them, like the fungicides, will obviously just impact it might have an effect on the fungal population. However, some pesticides actually act as a source of nutrients for microbes. So the microbes in the soil that are able to feed upon and break down these pesticides, we begin to see enrichments of these microbes. However, we can say that there is a shift in the microbiome of soils when pesticides are used. We see an enrichment of some and a decline in others. So it's kind of a yin and yang effect. When we think about this, we really need to focus on the microbes that might be important for crop production, like the nitrogen fixers. What are the pesticides doing to that? So I think there's a lot of work to be done, but a lot of people are working in this space. And so again, I would focus on the type of pesticide that it is, and it's not going to kill all the microbes. Again, it's going to enrich for many of the microbes. And these microbes we can also use in bioremediation. Now, when we move to the gut microbiome, that's, Seth showed that nice little image of how the microbes are fluctuating throughout the environment. So if you think of the soil microbiome being a little bit disturbed, well, that's also going to impact the flow of those microbes to the gut. We do see some residues of the pesticides in human urine. So we do know that it is impacting the gut microbiome. One particularly really active field of research right now is the gut-brain axis. And so people are just like really beginning to heavily look at the influence that pesticides might have on the gut microbes. So is it impacting the gut function? Is it impacting the way that these microbes communicate with one another or communicate with their host? And in doing so, can that pesticide then interact with the signaling between the gut microbes and the brain and the nervous system? So I think this is a very ongoing area of research. I would love to see students really get involved to help us detail the soil microbiome. And I really want to put a plug for the Penn State Microbiome Center, which has a number of soil microbiologists in it that are looking at these types of questions.
Thanks, Sarah. You reminded me that when those first cases of soil breaking down pesticides, a lot of it was about herbicides.
Mhm.
The first thought was that the weeds had evolved resistance to pesticides, and it took a while to figure out it was actually the microbes breaking down the herbicide in the soil that was reducing weed control.
Yeah, it's all about those interactions between the host and the microbe. Yeah.
You have to check carefully to get the right answer. How much, Seth, how much of the microbiome in humans is affected by food and diet? If I go get those tests done in 2 different days after I've eaten something different, will I see make much change?
It's possible. So I actually changed my diet for a year and a half to become a vegan because the microbiome sciences had a lot of data that suggested if you eat a vegan lifestyle, your microbiome diversity goes way up. Lots of leafy green vegetables is kind of the single recommendation one would give for diversifying your microbiome, where diversity tends to be better than not. If you eat a carnivore diet, a Western diet, high fat, high protein, high cheese, you actually have lower on average microbiome diversity than eating a highly vegetable or vegan diet. Now, the health status of that is, again, it's sort of the outcome is personalized to what conditions we experience in our lived lives. But it's clear that microbiomes help digest food. They help break down lactose in milk. They can help break down fiber in vegetables. They help actually convert meat into small metabolites. Now, here's a little bit of a concern if you're a heavy meat eater. So red meat is converted to a small chemical metabolite called TMAO by gut bacteria. And it turns out TMAO is a better predictor of cardiovascular disease than the canonical cholesterol that we all get measured in the doctor's office. This is new science from the last 5 years, really, that's telling us the way gut microbes process our meat, and that influences our risk for cardiovascular disease in a better way than we've traditionally measured. So general recommendation, just to sum up, would be to eat more vegetables, as our mothers and grandmothers told us. It really does have an impact on microbiome diversity, and the risks from eating red meat can actually come from the way your microbes break down that red meat. So things and food for thought, no pun intended.
Well, a related question that came up in the Q&A was, how do broad spectrum bactericides like hand sanitizer, mouthwash, chlorine, and other bactericides
Right.
in our passion to remove all the bacteria from our food systems impact our microbiomes?
Isn't it funny that we take probiotics and yogurts from TV commercials that we see every day and try and diversify our microbes from probiotics, but yet we clean our hands with sterilizer? And what's the rationale for that? Why are we choosing to diversify our guts but clean our hands? And why not treat these systems differently? The truth is, we don't have a ton of information on how these types of things that are not FDA regulated influence our microbiomes. Over-the-counter probiotics aren't even FDA regulated. So you may not really be doing what you think you're doing when you purchase that probiotic at a CVS company. In terms of the influence of antibiotics on our gut microbiome, it's clear if you do recurrent sampling of antibiotics throughout your life, you're actually taking hits at key microbes that get lost over time. And your body doesn't personally recover a small fraction of those microbes that get hit by those antibiotics. Now, that can lead, especially in childhood, to increases in inflammatory bowel disease risk, because what's happening is antibiotics ingested are lowering your microbial diversity time after time, and then your gut ecosystem is not healthy at that point. It's not diverse. So there is a risk to overdoing these kinds of taking in these kinds of molecules that will kill off our resident bacteria. We should think about that when we clean our hands too, whether that's good or bad, and that's still to be worked on a bit.
So another submitted question, Sarah, was about commercial brand boxes of tea sold in grocery stores touting probiotics or immunity support ingredients. Are they safe and effective? Are they regulated or commented on at all by the FDA? What do you think?
Yeah, I think, and going back to what Seth was saying, you know, all probiotics are not equal. So when I personally when I'm looking at a product in the store, I look at the label, just like you would look at the number of calories and the fiber and the carbs. You want to look at the ingredients and the types of microbes that are being added. So that cocktail of probiotics. Just because it's a microbe doesn't mean that it can survive through your GI tract. It doesn't mean that it's actually doing something beneficial once it's in your gut. As far as teas, I don't think teas naturally have probiotics. And so unless it's a fermented tea like kombucha. So the teas that are in bulk probably won't have a probiotic impact for you, but the ones that are being sold that are commercialized as probiotic, they often contain Bacillus coagulans, I believe, which is the same microbe that you'll often see in infant formula. So the FDA has determined that it's generally recognized as safe and effective. So it'd be comparable to taking a probiotic pill. However, again, when you're looking at probiotic things in the supermarket, I would again look at the label and maybe just generally Google that against the FDA to see what the FDA said about that particular microbe.
So a couple of the other big overarching questions. Does an individual's human microbiome have any epigenetic effects on their body or the development of a young child?
I can take that one because it relates to this hypothesis we talked about in the slide deck about early life influences kind of having an outsized effect on our health later on. One example of that would be, let's say, children raised on farms or children raised in houses with multiple pets, not just one, but multiple pets, tend to have lower rates of allergies and asthma. And the thought is that those children are growing up in an environment where they're more exposed to microbiomes. They're playing in the dirt. They're interacting with these animal pets or animals on the farm than somebody who's living in a household for most of their childhood life. So that tells us that the way we grow up can potentially influence the risk of chronic diseases later on. And there's very likely a microbiome by immune system training crosstalk that's being handled in those outcomes. Another example is babies born by C-section have a different microbiome when they come out rather than babies born by the normal route of a vaginal birth. And that makes good sense. A vaginal birth delivery, you get vaginal microbes on the baby, and a C-section baby, you get skin microbes. Now, C-section babies have also a much higher rate of allergies and asthma than vaginally delivered babies. And the thought is that there may be some microbes that are sort of miscolonized from mother's skin through a C-section rather than the evolutionary normal route through the vaginal canal. And that sets up some altered training of the immune system that relates to this concept of having the right microbes early in life could influence your risk of chronic diseases later in life. Those are the effects that, and some data that certainly suggest we need to think harder about what microbes are missing from what we do and how we live our early life experiences.
So another question was, what's the relationship between microbiomes and cancer? And that it must be incredibly complex.
Okay, so this is one of the most exciting areas of microbiome sciences, in my opinion, in the last 3 years, because there's been a flurry of data that has shown us clues that cancer is and has a microbial basis. And what's the evidence for that? Well, there's evidence that there's intracellular, so within tumor cell, microbiome communities. And each tumor type, whether it's lung or gut or skin, each tumor has its own specific signature of a microbiome community. Changes in those microbiome communities within the tumor cells appear to correlate with the progression of cancer and whether they become metastatic or benign. And if that's not enough, it turns out that the other side of this, treating cancer, let's say with chemotherapy, the effectiveness of that chemotherapy depends upon what gut microbes we have, because they're metabolizing the chemotherapy in our bodies are sort of taking that medicine, and they're not treating it equally if you have different microbiomes within your body. So the ramifications for cancer efficacy, development, and treatments are profound, but we're just scratching the tip of the iceberg in the last few years. And I think we're going to see diagnostics and treatments in the next decade related to cancer microbiomes.
Thanks a lot, Seth. There's one other questions we put in the Q&A, even though we're running out of time, I just have to grab this one.
Yeah.
So this was, what measures are being taken to ensure there are not unanticipated cross-species infections that suppress insect species populations that are non-targets or beneficial? I assume this is about Wolbachia and mosquitoes and so forth, isn't it? I mean, some of these trials have already been done in the field, I think in Australia and Southeast Asia, and presumably they're looking for this kind of thing. But
yeah,
is it the host specificity of the Wolbachia that keeps them pretty much?
So there's even a more general answer to that, which is Wolbachia occur in 50% of the world's insect species. So even if it jumps, right, and we don't have evidence of that, but even if it did, this is sort of normal for Wolbachia, right? It's jumping around the insect tree of life all the time. This would not be abnormal. In the event of one transfer from, let's say, a mosquito with Wolbachia that we released versus the thousands of events that happen naturally, is a drop in the bucket for a concern. So far, nothing to worry about there. And if it does happen, it's kind of a natural event that contributes to the larger story of how Wolbachia move around insects.
Well, thank you very much, Seth and Sarah. We've actually kind of overrun our time already, but it's fascinating stuff. Really opportunities for people to get to look back at your website and so forth. So it's shown on the screen, but we've got some upcoming events. We'll be at Ag Progress Days with College Connections a bit earlier than usual. But Secretary Redding and I will do a session looking at challenges in agriculture. And then in September, I'll be joined by Dr. Ed Dudley, who's a professor of food science, and Jasna Kovic, professor of food safety, to discuss Bugs in My Food 2.0. So we'll look forward to seeing you at some of those upcoming events. And with that, thanks again to Sarah and Seth for a fabulous program today. Great job. And we'll look forward to seeing you all again the next time. Take care.